For 40 years, the intellectual energy of the Alzheimer's field has been spent adjudicating a question of priority. Most frameworks are built as a linear cascade, where a single first-mover molecule initiates a sequence of events that terminates in the death of neurons. But human autopsy data presents a persistent anomaly. Up to 30% of cognitively intact elderly individuals pass away with brains that harbor severe Alzheimer's range amyloid and tau pathology. These people lived without dementia, despite carrying the supposedly decisive molecular lesions. A single substrate model struggles to explain this resilience. It also leaves the disease's 50-year latency and its stereotyped anatomically constrained progression through the brain as unexplained structural features. These anomalies motivate a shift toward a systems model organized by time, anatomy, and cellular state. This approach traces the disease through distinct, mechanistically bridged phases of collapse. To maintain rigor, we will map this architecture using a 3-tier evidentiary scale. Solid lines mark Tier 1 established facts, direct primary evidence in human tissue. Dashed lines denote Tier 2 bridging inferences, mechanistically constrained connections between known facts, and dotted lines represent Tier 3 conjectures, coherent hypotheses offered for future testing. Grading the theory joint by joint allows us to test the architecture at its connections, rather than merely accepting it at its edges. This schematic shows how the architecture accommodates existing single-molecule theories as parallel entry lanes. In this model, they are specific starting points that converge on a shared downstream pathway. In autosomal dominant Alzheimer's, amyloid beta is the initiating cause. For these patients, the disease begins as a Tier 1 established reality of amyloid dyshomeostasis.
The ecological lane starts with the Tier 1 fact that age-related dysbiosis depletes protective butyrate-producing bacteria. This depletion weakens the integrity of the gut barrier, establishing a permissive, low-grade inflammatory tone in the periphery. A Tier 3 inference then proposes that functional bacterial amyloids, such as Curly, cross-seed host tau proteins, transmitting a pathological signal to the brainstem through the neuroepithelial circuit of the vagus nerve. Whether a patient enters through a genetic, vascular, or ecological lane, the pathology eventually loads a specific anatomical node, triggering the first phase of the architecture. Phase 1 begins in the locus ceruleus, a small brainstem nucleus. Brock and Del Tredizi's staging provides the Tier 1 anchor. This is where the earliest detectable tau pathology appears, often decades before the first symptom of memory loss. The locus ceruleus neuron is defined by its metabolic extravagance. It sustains a vast axonal tree across the forebrain through autonomous pacemaking. This constant activity keeps the cell perpetually near the ceiling of its mitochondrial capacity, enduring a relentless oxidative load. Chronic oxidative stress causes DNA damage, which hyperactivates the repair enzyme PARP1. This process consumes NAD+ at such a high rate that it creates a bioenergetic sink within the cell. Depleted of NAD+, the cell loses the ability to maintain mitochondrial quality control. It can no longer clear depolarized mitochondria or synthesize the components needed for repair. Instead of undergoing immediate somatic death, the neuron enters amitosenescence, a stable, dysfunctional state where it persists while secreting inflammatory factors into its surroundings. While this senescence program is established in dopaminergic neurons, its specific role as the initiator of Alzheimer's remains Tier 3 conjecture.
The disease takes its specific form because this generic engine of cellular aging acts upon the unique selective vulnerability of this exact neuron and its specific anatomical wiring. The architecture of the disease begins with this silent, decades-long metabolic crisis deep in the brainstem. For 30 years, this bioenergetic failure is a private affair of the brainstem, but the disease eventually crosses a physical bridge into the forebrain. The bridge is the locus coeruleus's own axonal tree. This projection system is one of the most extensively arborized in the brain, broadcasting signals from a small brainstem origin to the entire forebrain. These projections carry two distinct cargoes.
The first is the transsynaptic seeding of misfolded tau, which templates pathology into the hippocampus in a prion-like manner. Simultaneously, the degeneration of these axons leads to the withdrawal of noradrenergic tone. In health, noradrenaline suppresses pro-inflammatory transcription in microglia, acting as a standing brake on neuroinflammation. This Tier 2 dual cargo convergence, pathological seeds arriving just as the inflammatory brake is removed, converts a metabolic failure into a regional immunological crisis. Phase 2 is the collapse of microglial homeostasis in the limbic system, triggered by the arriving tau seeds and the loss of noradrenergic suppression. Departures from the microglial homeostatic signature are often described as steps down a single degenerative slope. However, the data reveals a branched state space. Activated microglia can follow several different physiological trajectories, and they are not all harmful. Tier 1 findings show that certain activated states are pathology restricting. These cells remain clearance competent and actively work to contain the spread of amyloid and tau. But chronic stress eventually drives the system toward a terminal senescent detractor. These microglia become dystrophic and gorged with lipids, losing their ability to perform housekeeping functions. In this specific terminal state, clearance failure and active harm become the same cellular act. The cell that can no longer clear proteins begins to actively secrete enzymes that attack the surrounding architecture. Grouping all immune activation into a single category obscures the precise transition into the senescent state that executes cortical demolition. In Phase 3, these senescent microglia converge on the parvalbumin interneuron. This cell is the primary metronome of cortical computation, generating the gamma rhythms underlying working memory and attention. Parvalbumin neurons are physically encased in the perineuronal nep, a dense extracellular matrix that serves as an ion exchange buffer and an antioxidant shield, protecting the cell from the stress of its own high-speed firing. During the proteolytic turn, lipid-laden microglia switch to secreting matrix proteases. This transition initiates the rapid digestion of the perineuronal net. This Phase 3 bridge is a Tier 2 multiplicative attack. The net is fragmented by proteases, weakened by iron-driven Fenton chemistry, and marked for microglial stripping by the complement system. Stripped of its shield, the parvalbumin neuron does not immediately die. It falls electrically silent, down-regulating its activity to survive the oxidative load. The consequence for the network is a loss of perisomatic inhibition. Without their pacemakers, surrounding neurons fire wildly, and the gamma rhythms cognition requires collapse into hypersynchrony. This physical disconnection of inhibitory circuits at the perineuronal net is the mechanical reality of dementia, often occurring independently of the total plaque count. This bottleneck explains the resilience paradox. It explains how 30% of people with high pathology brains avoid dementia. This separates the resilient brain from the demented. Both are saturated with plaques, but cellular states differ. Resilience preserves homeostatic microglia in pathology-restricting branches. Because microglia remain stable, the perineuronal net survives. The parvalbumin interneuron stays shielded, pacing the rhythms that support cognition. Dementia tracks with the severing of connections at the matrix, not the gross accumulation of lesions. If the inhibitory scaffold is preserved, cognition remains intact. This architecture suggests a mechanical reason why single-target monotherapies have historically failed. A static tool cannot address a phase-shifting, multi-lane process. Asecta's intervention requires matching the strategy to the phase: metabolic support in Phase 1, keeping microglia homeostatic in Phase 2, defending the matrix in Phase 3. Applying an upstream custodial fix to a downstream structural collapse is futile. Likewise, clearing all microglia indiscriminately risks removing protective cells before senescent ones take over. The validity of a systems model rests on the strength of its structural joints. To treat the disease effectively, we must lend to see the pathology's front while it is still upstream and match the tool to the specific phase and the specific patient.